Perfect. I'm just gonna turn off the camera because my internet is just acting funny. Well, thank you, Jamie, for the introduction. And I just want to start by acknowledging the traditional owners of where I stand today, the Kaurna people, and recognize their continuing connection to land, water, and culture, and also to pay respect to their elders, past, present, and emerging. So the study that I'm going to present today is focused on the Gippsland Lakes. This is the largest estuarine coastal lagoon system in Australia, which is located in the state of Australia down here. And this system consists of a complex network of channels and water bodies interconnecting 5 major rivers, which flow into three main lakes, which is Lake Wellington on the east, then Lake Victoria in the middle, and Lake King in the west. This system hosts a diversity of wetland with high conservation value. However, over the last 2 centuries, the Gippsland Lakes have suffered a substantial modification, mostly associated with anthropogenic impact, like hunting, introduction of exotic species, deforestation, and one of the most significant changes made to this system was the permanent opening of the Gippsland Lakes in the 1889 after the European settlement, where an artificial channel was made connecting lake entrance to the Tasman Sea, which looks like that. And this permanent entrance has generated several environmental problems over the years, resulting in, of course, an immediate change in water levels in the system and a chronic salinization of a formerly fresh to brackish water system, which of course affected the local biodiversity at pretty much all ecological levels. So Lake King and Lake Victoria were the most affected by this channel because they were much closer to the opening, and Lake Wellington was less affected by this. As you can see here in this image, the bottom right, the color code indicates the salinity concentration in the system, going from low salinity in a green color to higher salinity in red. So you can see that here, Lake King and Lake Victoria suffer the most with this opening of the system. So we knew from previous paleo studies in this study that the anthropogenic impact created periods of eutrophication, but there was still little known about how other species responded to these historical and human-mediated changes. And for this study, we specifically focus on prokaryotic communities because microbes can be a powerful proxy that can be used as an indicator of environmental response. And the few studies developed in this system previously have mostly used classical microbial paleo indicators like diatom, identified from subfossils and pigment. However, we were still missing most of the identification of the microbial communities here. So in this study, we investigated a multi-decadal scale changes in prokaryotic community structures using sedimentary DNA. And for this, we took sediment cores from Lake Victoria and Lake King and tested if the anthropogenic impact of the European settlement could be detected with microbial sedimentary ancient DNA specifically. So for that, we extracted DNA using a silica-based methods for ancient DNA. And then we used shotgun metagenomics to track these changes in past ecological communities, and then correlated these changes to nutrient levels pre and post European settlement. So once we dated our sediment samples to know which core depth corresponded to post-European sediment and pre-European sediment, the next step was to assess the historical changes in prokaryotic communities, was to analyze the nutrient levels along the sediment course to understand how those long-term alteration affected in the sediments. And for this, we use stable carbon and nitrogen isotope and also carbon to nitrogen ratio. So here in orange, we have the nutrient levels from Lake Victoria, and in blue, Lake King for the carbon nitrogen ratio and nitrogen isotope and carbon isotopes. And we can see that there is an overall increasing trend for both Lake King and Lake Victoria of those nutrient levels in post-European settlement. However, there was a few fluctuation in pre-European settlements. It wasn't as clear as we thought, but one of the nutrients level that really show this change was nitrogen. It had a more clear change, in this case in Lake King after European settlement, and also in Lake Victoria after European settlement. So overall, we could see a nutrient enrichment in the Gippsland Lake associated what we think is with anthropogenic activities from European sediment. So now that we established those changes in nutrient levels, we wanted to investigate the changes in the prokaryotic communities in these periods. But because we were interested in past communities, the first thing that we wanted to examine was the DNA fragmentation and damage along the sediment, as both of them are the two main characteristics of authentic ancient DNA. So for the DNA damage analysis, we mapped our reads to DTDV database, which contains bacterial and archaeal genomes. And then we screened for sequences with signs of increased deamination rates at both ends of the reads using the software hops. And we wanted to start with that, as I mentioned before, because we were focusing on past communities. And so the first thing that we wanted to see is that if we actually were extracting ancient DNA. So for the analysis, we were expecting to recover a higher number of damage and short reads at deeper layers of the sediment, so here at the bottom, as they represent older samples. However, from the fragment length distribution plot on the left, we observe a high variability in fragment length along the sediment course in both lakes, especially in Lake King between around 100 centimeters and 150 centimeters. And of course, we didn't observe any significant correlation between the fragmentation rates and depth or age of the samples in any of the lakes. And for the DNA damage analysis on the right, we use the percentage of damage reads from the total number of reads that were mapped to this database in each samples in both lakes. And so again, we were hoping to see that deeper layers should contain a higher percentage of damage. And for this analysis, we did see an increase of reads that were showing damage in Lake Victoria, starting from around 150 centimeters. But for Lake King, we saw the same trend that with the fragmentation, these layers here between 100 and 150 were actually showing a decrease of damage compared to the previous samples and samples after that. And again, there was no correlation or significant correlation between DNA damage and death in these lakes. So, So because one of the reasons we thought that this could be happening is that because sedimentary microbial communities are a mixture of modern and past or ancient species, we could actually have a higher contribution of modern living prokaryotes at this core in Lake King at this depth between 100 and 150 here. We could, in a way of trying to separate past communities from the modern communities, we separated the reeds from each sample FASTQ file according to the fragment length. And we placed them into 50 base pair fragment bins. So now for each sample, we had six FASTQ files and containing the separate fragment. And we did this again, the same analysis with hops. So the idea was that we have the mixture of different fragment length of DNA. And because ancient DNA is often much smaller in length than modern DNA, we wanted to separate those smaller fragments into different bins. And again, as I mentioned before, we have here the different fragment bins, the different files that were mapped to the database and then screened for deamination rates with hops. And we had a look again at the DNA damage ratio in each of those bins along the sediment core. And we observed a few interesting things. So one, in average, reeds shorter than 100 base pair had slightly higher DNA damage percentage compared to longer reeds in both lakes. And the second observation was that there was a higher variability in damage in reeds longer than 200 base pair, probably because we were recovering a lower number of those reeds in these bins, but they could still introduce noise to the analysis when we were analyzing all the reeds together. So if you see here, the variability of the DNA damage seems to increase in longer fragments in both lakes. And but the reeds from Lake King was still showing a high variability in their DNA damage. So we couldn't observe any correlation with depth in this lake. But in Lake Victoria, now that we separated the fragments, the shorter reeds here from lower than 50 base pair and lower than 100 base pair, did show a positive correlation now with depth compared to the rest of the bins that did not show a positive correlation with depth or the age of the samples. So overall, we can see an enrichment of authentic ancient DNA molecules in short fragments, at least in Lake Victoria, especially at deeper layers of the sediment core. So again, now that we confirmed that our samples contain authentic ancient DNA signals along the sediment cores, the next step was to examine if any of the variables that we had, like depth or fragment size in this case, the European sediment or the neutron levels, had any association with the changes in prokaryotic compositions and abundance. And for this, we use the map reads from the previous analysis, and we assess the beta diversity of the samples and with Bray Curtis, and visualize it by non-metric multidimensional scaling plots, and then use PermaNova to identify significant variables. And here are the ordination plots where samples are shown in different colors. And the samples from pre-European sediment are circle in red, and the post-European samples are circle and blue. Figure A corresponds to Lake Victoria and figure B to Lake King. So the first obvious thing that we noticed is that the fragment size was not a significant driver of the composition in neither of the lakes. So the bins were actually clustered mainly by samples and not by the fragment size. And in Lake Victoria, figure A, there was a clear and separate cluster between pre and post-European sediment periods, and also increases of carbon to nitrogen ratio and also nitrogen isotope levels were significantly correlated with samples in post-European periods. There were also younger samples, suggesting that European sediment was strongly affecting the vertical changes of these communities. And this observation was also confirmed by an Adonis analysis, where European sediment was the most significant driver of the microbial composition, explaining almost 40% of this variation. So in Lake King, on the other hand, the separation between pre and post-European sediment was not as clearly defined as in Lake Victoria, mostly because some of the samples, especially those two samples that, if you remember before, had this issue with the fragmentation rates and the DNA damage analysis, were correlated and clustered with, again, younger layers here. And we're also significantly correlated with an increase in nutrients levels like carbon and nitrogen ratio and nitrogen isotope. So there was something definitely happening and changing in this layer, and it was probably explained by the nutrient inputs of the catchment during these periods. So the next step that we wanted to do, the final step, was to actually investigate the specific changes in the prokaryotic communities in these periods to see what was exactly changing along the core. And here is a taxonomic plot at phylum level showing the vertical microbial composition of the sediment cores from Lake King and Lake Victoria, showing also the composition of each of these data sets separated by fragment length. So we have here pre and post European periods, the depth of each of the samples, and which fragment bin corresponded and their composition. So again, the first obvious thing is that both lakes presented shift in their several taxa along the sediment core, and especially after post-European sediment. And between the most significant changes, we found that in both lakes, taxa belonging to the phylum Proteobacteria, you can see here in dark green, were more abundant in upper layers of the sediment in post-European periods. So most of the species that were more abundant in this phylum was belonging to the order the Sulfobacterales, which is an order of sulfide-reducing bacteria. They can obtain energy by oxidizing organic compounds, and its high abundance has been positively correlated with organic-rich sediment. Thus, that increase that we're seeing in both of our lakes in the data sets could actually reflect a period of eutrophication in post-European sediment, which we also observed in our nutrient analysis, and there was previous records of that, especially terrestrial organic matter in this lake. In Lake Victoria, on the other hand, on the left, taxa belonging to the phylum Candidatus Batearchaeota in light orange this over here, and Candidatus thoraciota here in red also show a significant increase in their abundance in post-European sediment. And Batrachiotta species are usually highly abundant in anoxic sediment. And again, this enrichment in post-European sediment could reflect a decrease in dissolved oxygen in Lake Victoria, which was also previously recorded in other studies, probably due to the large increases in nutrient inputs, which has been identified as important drivers for the formation of hypoxia in lakes. And in Lake King, we also observed that the core depth that presented less DNA damage and fragmentation show an increase of the same species, the Sulfobacterales order, and also Agribacteria, which is commonly found in anoxic sediment. So we think that these species could be modern living organisms that are thriving in these layers, mostly because of the increased input of nutrients. And finally, one of the major findings in this analysis was that although the analysis from the ordination didn't show a clustering according to the fragment size, we did find some changes in the taxonomic composition between different fragment bins. So for example, a species from the phyla Plactomycetes, Chloroflexi, and Thoraciota were significantly more abundant in bins with shorter fragments. So this could represent past communities, but of course we cannot rule out that this can have different degrees of DNA preservation compared to the rest of the species. And this is actually something that we want to explore, and we're exploring now. So I'm just gonna go to the end, sorry. As a summary, our results show that there are vertical changes in sedimentary microbial communities correlated to nutrient levels, core depth, and European sediment. And these findings suggest that the prokaryotic communities likely responded to both past environmental conditions and also geochemical changes of the sediment during burial. Also, the microbial communities identified from the DNA preserved along the sediment course in the Gippsland Lake show differences in their DNA fragmentation and DNA damage between taxa, suggesting that there was a mixture of modern and ancient species. And these observations suggest that these communities could be useful paleobioindicator of historic management practice of waterways. Thank you. Hi, can you hear me?
Okay, thanks for that, Vilma. Does anyone have any questions? You can either just call out questions or write them into the chat box. I've got one, Vilma. Yeah. Just out of curiosity, what was the original purpose for creating the connection with the ocean?
So there was ships coming into the system lake. So they started to do some dredging of the lake, but apparently they needed to, because there was so many ships coming in and out of the system, they just expanded the entrance basically. Mm-hmm.
Yes, I have two quick questions. Really cool talk. Thank you. First, I think I missed it. What was the time span of these cores?
At around 200 years.
Okay. And oh shoot, I just forgot what my second question was. It will come back to me. Oh, I remember, sorry. So is there any plan to try and look at any of the geochemical, like any geochemical signatures or anything to see if that might relate to some of the observations that you have?
Yeah, so there are some, we're actually waiting for the results, but it's taking a really long time. So we decided to just move forward with the study without those, but we did send samples to have those analyses.
Cool, that'll be really exciting to see.
Yeah.
Vilma, I was just going to ask about your taxonomic plots. The one interesting thing that stood out was, I think in Lake Victoria, the nitrospirane seems to disappear at the time of European settlement.
Sorry, can you repeat that?
The purple, is the purple?
Yes.
Sorry, the nitrospirane.
Yeah.
They seem to disappear at the time of European settlement.
Yeah. So we haven't had too much in depth into that group. We're still going through a lot of that because the changes for this community in Lake Victoria, there was actually a lot of species involved there. And I don't think nitrospirine was one of the most significant ones. So I couldn't really say much about it, but mostly because we also did the analysis not at phylum level, but at the species level of this phylum. So it wasn't really significant when we had a look at that in the end.
Okay.
Hey Vilma, I've got a quick question.
Yeah.
I can't remember the first sort of, sorry, I have trouble talking, I'm coughing all the time. The first slides that you had about the locations of your lakes, does it make sense basically based on the locations that you see these big differences between the lakes?
Yes, yeah. Sorry, I think I couldn't hear you very well. Could you repeat the question, Linda?
Yeah. So you see these big differences between the lakes, basically, with Lake Victoria has a super clear signal in changes in communities.
Yeah.
And Lake King is a little bit less, I guess, obvious. At the beginning, you had, I think, a map of the different lakes and the settings.
Yeah.
And does this make sense in terms of a location point of view of where the lakes are?
Yeah. So
changes.
Yeah. So it is a big factor. So at the beginning, I actually thought that all of these changes, like very clear changes, I was gonna see with Lake King because it was like directly connected to the ocean and had the major changes in salinity. But we also discovered that there was a lot of activities around that catchment closer to Lake King. So the input of organic matter in Lake King was much higher than Lake Victoria. And they were doing a lot of dredging too, closer to Lake King. And I think that was just kind of moving the sediment, if that makes sense.
Yeah. Thanks. Interesting.
And we've just got a question in the chat from Lucinda. So just, Vilma, just on what you were saying about the preservation of some ancient taxa being better than others.
Yeah.
What do you think are some of the major reasons why some prokaryotes would preserve better than others in your sediments?
So I didn't have a look at that directly, but other studies have tried to correlate, for example, GC content of the species with the preservation that the DNA could have. And from the species that were significantly higher in shorter fragments, they actually had higher GC content. So we think that it could be that. Actually, ancient species that we still are able to see and extract their DNA, basically because their preservation was better compared to other taxa. But other than that, I mean, it could be several reasons. It could also be the temperature, or how long they were living there, because we also found damaged reeds. I mean, we found damaged reeds from the species, but we also have non-damaged reeds from the species there. So it could also mean that they're actually living there, but at slower rates. So there's a lot of things that we still need to test, but we just don't think that we have the data in this study to do that. So we are doing that with other sediment cores from Australia. Does that answer your question, Lucinda?
Can I ask a stupid question? What's GC content?
GC. Sorry, GC.
What? I still don't know.
the content of in the genome, cytosine and guanidine.
Oh, GC. Oh, I see. Yeah. Sorry. I was like, what does that stand for? No, all good. Thank you.
I have a question for you. First, great talk, thanks. And I have a question about your sequencing. You said you use a shotgun technique.
Yes.
And I was wondering if you did a PCR targeting the 16S region before the shotgun, or you took your DNA and you did the shotgun right away.
Oh, we did it right away. We usually do it like that, and we have never had any issues with the sequencing. So I don't know. I know that, for example, Linda, that is also here, she has worked with older samples, and I don't know if maybe she does something like that previously. But because of my sample are not that old, I usually don't need to check. They are fine.
Okay, thank you.
Cool. Do we have any final questions? Okay, well, I'd just like to thank Vilma for her talk today, and thank everyone for joining in and listening. We'll make this a regular thing, probably once a month, with a speaker from this part of the world. And so we do have a couple lined up. We've got one for next month, September, October, November. So at least the next few months, we've got speakers sorted. But if anyone is keen to give a presentation, just flick me an email and we can pencil you in for later in the year. So yeah, thanks everyone, and we'll end it there.
Thank you.
Thank you very much.